So cell division occurs differently in between prokaryotic cells and eukaryotic cells, mainly because those prokaryotic cells lack a nucleus. So there is that single circular chromosome that's just floating in the cytoplasm. So it's in the form almost like chromatin in a way, where it's just that circle is just balled up on itself. It's very unorganized, and that is one of the reasons why bacteria can evolve so quickly and they adapt. So the process that they use is called binary fission. It's the process of making extra organelles, duplicating that chromosome, and then splitting into two identical cells. So I have an example here, and my picture is similar to yours but not identical. So when we look at the first stage right here, the cell basically elongates while the DNA is being replicated. Because of course, if there's not two sets of DNA, you can't produce another cell. So in that nuclear area right here, we see two pieces of chromosome that are being duplicated. And then we can see right here what's called the cleavage furrow start to form. And that's where the cell wall and the plasma membrane are being built from the outside in. Now once those two sections meet, once it's completely formed, it completely encompasses the two separate pieces of chromosome material, and then it can entirely separate. So what we see right here in this electron micrograph is we can see that beginning of the cleavage furrow where there's that kind of that cross wall is just partially formed right there, and we can see the chromosome, that genetic material, separated into two different cells. Now in eukaryotic cells, the cell cycle occurs very, very differently. And the cell cycle involves two different processes depending on which kind of cell is dividing. So remember, you've got somatic cells, or those cells that make up all of your body. So your skin, your blood, your bone, your muscle, your nervous, okay? All of those kinds of cells go through a process that involves mitosis. And mitosis is nuclear, it's actually nuclear division, in which the chromosomes of the original cell are copied and divided into two new cells, okay? Now, remember that in sexual reproducing organisms, there are gametes. So in females, the egg, and in males, the sperm. Now, if those two cells came together to fertilize and become a zygote, or the first cell that becomes a new organism, if our diploid number is 46, and those egg and sperm each had 46 and came together, whew, boy, that would make 92 chromosomes. Way too many, okay? Too many chiefs in the tribe, so to speak. So the cell wouldn't know what to do. So meiosis is the process of dividing the genome in half to make reproductive cells, okay, to make those haploid cells. So the idea is we're going from 46 chromosomes down to 23, okay? So keep that in mind. The cell cycle, cell division in eukaryotic cells that have a nucleus, is going to go through one of two processes. The cell cycle including mitosis, or the cell cycle including meiosis. So let's take a look at the cell cycle itself. It's a series of events from mitosis or meiosis to another cycle, okay? It repeats itself. So interphase is basically the first phase. It is a period of growth and development between cell divisions, okay? It's a little bit more complex than that, but we're going to get to that in just a second. The second part of the cell cycle is the M phase, and this is the reproduction phase. Mitosis, M stands for mitosis or meiosis, and that's the division of just the nucleus. So once the nucleus divides, then we got to get the whole cell to divide. So the third and final stage is what's called cytokinesis. Now that prefix, cyto, right there, refers to a cell, and kinesis is division. All right? So when we take a look to your right in your packet, you've got this diagram, and see if you can point out where exactly does the cell cycle begin. Well, you might be looking for one of two things. You might be looking for interphase, because interphase is the time when the cell is growing, so that would mean it's a new cell. Or you might be looking for cytokinesis, and cytokinesis indicates where the cell divides and becomes two new daughter cells. So that actually happens right up over here. So you might want to put a star right there in that area, right in between cytokinesis and what's called the G1 phase. Now, on this inner circle right here, and you may want to go back and almost color code this with markers, okay? That actually would not be a bad idea. This first phase right here, this is interphase, okay? So interphase is divided up into this green, yellow, and blue sections. G1, or what we call cell growth, and we're going to write these down on the next slide, so don't worry about writing them down yet. There's the S phase, or the synthesis phase, and then there's the G2 phase, which is the preparation for mitosis. You can see that this external piece of the pie here is what we would consider mitosis and then cytokinesis. So mitosis is actually divided up into what we call prophase, metaphase, anaphase, and telophase. Then there's that splitting of the cell, and the process starts all over again. So let's write this down. In interphase is divided up into three basic stages. G1, or growth 1, is when the cell doubles in size and the number of organelles. You can't just take a single cell, split it in half, because if it continued to reproduce, eventually that cell would have nothing in it. So you have to make more in order for there to be enough sets of everything, enough mitochondria, enough ribosomes, enough cytoplasm for two cells to actually be produced. The S phase, or synthesis, is when the DNA replicates, because of course you can't reproduce if you don't have an extra copy of DNA. And then there's G2, or that preparation, getting ready to reproduce, or to split in half. And that's when the enzymes and structures for mitosis are actually made. Now, some cells will actually exit the entire cell cycle and go into what we call G sub 0. So this spot right here, put a little star right there, because this is a really special time for a cell. When a cell exits the cell cycle after G1, DNA is not copied at all, but the cell is fully mature. So you've got a lot of body cells that are actually like that. Can you think of any cells that once they form and are functional, don't reproduce? If something gets happened to them or damaged, you can't get them back. Can you think of any like that? Probably the most common would be nervous cells in the spinal cord or in your brain. Once you damage them, you can't ever get them back. They exit the cell cycle, they don't replicate themselves, so therefore they can't replace any dysfunctioning cells. So, like I said, that's kind of an interesting time. Another time is, for example, red blood cells. Red blood cells are produced and only have a life of like 41 days. So at the end of that time period, those cells literally die and new ones are made in your bone marrow. So just an interesting idea. So they would exit from G1 to G0 and never go through S or G2, never even finish interphase. Now, the next phase is called prophase. Okay, so we're starting with I. P, okay, is for prophase. This is when the chromatin coils up into those chromosomes and we can actually see them. This is the first time that we can actually see chromosomes. You can see that the nuclear envelope and the nucleolus start to disappear, and then the centrioles begin to appear in animal cells and make themselves visible. Don't worry about what it looks like. We're going to look at a diagram on the next page. Well, on the back side of your notes anyways. So metaphase is the next phase. Now meta, this prefix right here, means middle. You might want to jot that down. Meta means middle. You can see that this phase is probably the shortest phase because it's only visible when the chromosomes literally line up in the middle of the cell. And what I want you to think about is that if the cell has two poles, like a north pole and a south pole, the chromosomes are literally lining up on the equator. Now, anaphase is when the centromeres are broken and the spindle fibers that have grown out of those centrioles, literally, which have attached to the centromeres, pull those chromatids apart to opposite sides of the cell. Telophase is when the chromosomes uncoil into chromatin and the nuclear envelope and nucleolus reappear, and then the centrioles disappear. Telophase is the exact opposite of prophase. You might want to write that down up here, oops, right here next to telophase. Telophase is the exact opposite of prophase. At that point, usually during telophase, you can start to see that cleavage furrow form, or that indentation in the cell membrane, which eventually ends up pinching the cell in half. So this divides the cytosol and organelles into two new cells. So during cytokinesis in plant cells, a cell plate forms between the two new cells. That cell plate will eventually become the cell wall. So that's what goes on your line right there. It eventually becomes the cell wall. Good, let's take a look at the picture. Well, we can take a look at this too. The cell cycle includes interphase and mitosis. Interphase is divided up into G1, S, and G2. This isn't in your packet, of course. And then the M phase, or mitosis, is divided up into prophase, metaphase, anaphase, and telophase. So if you would, just jot down this acronym, IPMAT, or IPMAT, IPMAT. If you can remember that, you'll be able to remember the phases of the cell cycle. Now, ultimately, if you can't, at the end, we have to remember C, because that's cytokinesis. So at the end of all that, then we've got the whole cell cycle can start all over again. Good, so take a look at the diagram that's in your packet. Mine is in color, and I want you to focus on what it is that you can see and how it might be the same or different than what's in your packet. So in this first page, there's interphase. The DNA is in the form of chromatin inside the nucleus. We can see the nuclear membrane, and right at the end of interphase, we can start to see those centrioles begin to form. Now that's not depicted in your note packet. But in the next phase, this is when the centrioles begin to form those spindle fibers, long microtubules that are going to attach to those centromeres inside the nucleus. As the nuclear membrane starts to break down, those chromosomes become visible as paired chromatids. Then, in metaphase, remember, meta-middle, so if we look at North Pole, South Pole, here's the equator. That's the only time you ever see those chromosomes all lined up nice and neat. In anaphase, the centromeres are broken and the chromosomes begin to pull apart towards those poles, getting equal amounts of DNA at each end, making sure that everything is there properly. In telophase, again, we can see that cleavage furrow starting to form right there. But now we can see, remember, reverse prophase. So the nuclear membrane is starting to reform. The spindle fibers have disappeared. These chromosomes are going to begin to unwind or uncoil and form chromatin. And then eventually, cytokinesis occurs, and I have two new daughter cells.